Hi, I'm Dr. Leslie Blankenship-Williams, and in this lecture, we are going to look at the historical perspective of microbiology up until about the 1800s. Now, from a previous lecture, we learned that microbiology is largely the study of microbes, and the most classic definition of a microbe is going to be a living organism that requires a microscope to view. So it would make sense then that the field of microbiology is rather new and didn't start until microscopes were able to view small creatures like bacteria. So when did that happen? Well, that actually happened in the 1600s. So the first person to view a living cell with a microscope was Robert Hooke, and in 1665 he made his discovery of the idea that cork, which is a plant material, was made up of plant cells. But remember, eukaryotic cells are big relative to little bacteria, so it actually required a better microscope to be able to view things like bacteria, which are much smaller. Turns out that the first person to do that was about 20 years later, a Dutch dabbling scientist, amateur scientist, you might say, someone who just kind of dabbled on the side, named Anton van Leeuwenhoek, was able to observe bacteria for the first time in the 1680s. Now, I'm going to put a pause on that concept for a moment and talk about this idea that, yes, microbiology as a formal discipline really started with the invention of microscopes that could see microbes. But of course, infectious diseases, which are caused by microbes, have been around for far longer. So really, the field of microbiology goes way, way, way back. In fact, as far back as humans have evolved. But they weren't known to be part of microbiology until the late 1800s, when the germ theory of disease was proved. And we'll talk about the germ theory of disease in the next lecture. So what did we know about microbiology before the invention of microscopes? Well, now that we know that infectious diseases are caused by microbes, we can turn to the annals of history and start kind of looking at, well, what did people observe and know about infectious diseases? And of course, there's a wide swath of infectious diseases out there, many of which are geographically restricted to certain areas like malaria, but some that were really well documented, particularly because of their catastrophic effect on the civilizations that they swept through. And this would include plague. It also includes syphilis and, to a lesser extent, leprosy. These are three that have been fairly well documented historically. So let's first talk about leprosy, because you see references to leprosy even in the Bible. Now, leprosy nowadays is a pretty unusual disease, but historically it was important, and we now know that it is caused by a bacterium by the name of Mycobacterium leprae, very slow-growing bacteria that causes some pretty grotesque and severe disfigurement, a very slow disfigurement. People that were afflicted with leprosy, which was incurable at the time, were isolated from the rest of the population. So there was definitely the idea of contagion. That is, someone with leprosy could somehow transmit it to somebody who did not have leprosy. And as a way to prevent that contagiousness, again, they didn't know what it was. They just thought something about this seems to be transmitted from person to person. They started isolating, starting like probably in the 1000s, maybe 1100s, they started isolating people into what are called leper colonies. So these are special areas where lepers are basically forced into exile permanently for the rest of their life, with the purpose of protecting the general population. And leper colonies actually existed up until, last century even. The last one in the United States was in Hawaii. And so leper colonies are an example of quarantine. The idea of quarantining infectious individuals to prevent the spread to the general population. And of course, if you've lived at all in this last year, you definitely know that word quarantine, even if you didn't prior to COVID-19. And so here we have an example of a now, well understood public health technique of quarantine to control the spread of disease. And I want you to remember that people did not know what caused leprosy. It was just this idea of contagion and this fad of putting them somewhere else and hoping that it didn't spread. Now let's turn and take a look at the effect of some of these diseases on life expectancy. In this table, look at the boxed area, and what you'll see is the importance of infectious disease in causing death. So even up until the 1850s, infectious disease was responsible for almost half of the deaths. Fast forward to more recently, first world countries with advanced medical care, infectious disease is responsible for a lot fewer deaths. Whereas in poverty-stricken countries, ones that don't have great access to medical care, infectious disease is still a prevalent cause of death. Now, the more common infectious disease is as your cause of death, the lower the life expectancy for the general population is going to be. And we see that in this chart. So you can see the overall life expectancy kind of hovers around 40 years of age up until the modern medicine. So when did we figure out that the microbes were causing diseases? The late 1800s. When were antibiotics introduced? The 1940s, really. And so you can kind of see these, like, we recognize that microbes cause infectious disease and we start to see an uptick. It's not the only reason why we have an uptick, better infrastructure, sanitation, etc. People weren't starving as much, blah, blah, blah. There's other reasons why we have an uptick, but that's one of them. And then antibiotics were introduced in the 1940s and you see an even greater uptick. And so now we have a pretty substantial life expectancy. So now that we've looked at the importance of infectious diseases historically, then it comes as no surprise why people were so interested in trying to figure out how to deal with them so that people didn't die. Now, one of the major catastrophic diseases that wiped through human civilization time and again, leaving a lot of death in its wake, is the plague. Now, the plague is kind of a generic name that actually might encompass a couple different diseases. So like the first plague, the Athenian plague, I think in the BC time, probably wasn't actually the same plague that we saw in the 1800s in London. But so plague is kind of this generic idea of something wiping through, an infectious disease wiping through civilization and causing a lot of death pretty rapidly. Most cases of plague, including the Black Death of the 1300s, is caused by a bacterium known as Yersinia pestis, and Yersinia pestis causes bubonic plague. So bubonic plague is marked by the formation of what is known as buboes. Buboes are horrifically and grotesquely inflamed lymph nodes. You know how when you go to the doctors and you might like have strep throat and they kind of feel here? They're feeling for lymph nodes, right? So if your lymph nodes are enlarged, that suggests a pretty serious infection. But rarely are they so enlarged that they stick out like golf balls like this, like they do in the plague. So they actually gave a name to these grotesquely sticking out lymph nodes, and they called them buboes. Now, buboes, or lymph nodes in general, tend to show up either in the neck, under the armpit, or in the groin, because that's where you have a large concentration of these lymph nodes. So the bubonic plague was absolutely devastating. Like in the 1300s, they think maybe even half of Europe was wiped out in a decade over this particular plague. So, of course, what were people doing about it? Well, people were trying the same thing that they were trying for leprosy. Run away. The rich ran away. Run away from crowded, civilized areas full of dead bodies. Go to the countryside and hopefully it won't be there. Well, it didn't really work. That quarantine, self-imposed quarantine, did not work. Well, it worked a little bit, but it didn't work to the extent that you would expect. And that is because humans do not readily transmit plague from one person to another. Rather, the plague is what is known as a zoonotic disease. Zoonotic diseases are going to be diseases that originate in another animal. COVID-19 is a zoonotic disease. So zoo, like going to the zoo, stands for animal. So zoonotic diseases tend to be pretty devastating when they cross over to humans for a variety of reasons. But plague is no different. The typical reservoir or area that plague, the bacteria that causes plague, likes to hang out is the black rat. And of course, in this time, rats are everywhere. Now, how do you get it from a rat? Are these people that are handling rats or something? And not necessarily. They are transmitted by fleas. So the bacteria actually bite the rats and then bite the humans and transmit it. Now, how the bacteria get the fleas to bite humans is really interesting because fleas, if given a choice between a rat and a human, are usually going to pick the rat. Fleas are not naturally found on humans most of the time. So how is it that humans were getting bit so often? Well, this is a sneaky little bacterium. When a flea bites a rat or a human that has Yersinia pestis, the bacteria gets sucked up into the esophagus of the flea and then basically cling on and form a seal, a plug of sorts. So a mucus plug up here in the esophagus. Uh-oh. So that means this flea, every time it eats, has a stopping measurement here. So every time this flea eats, tries to suck up blood, that blood is blocked from going into the digestive system by this esophageal plug that's just teeming with Yersinia pestis. So what that does is that does two things. First of all, that makes the blood regurgitate really quickly, and it basically takes the top layer of that mucus plug with it, which is teeming with bacteria. So if you are a flea and you draw up blood and then the blood just comes right back out of you, then that means that you're dropping all of that blood back into the bite wound with new bacteria. So that's how you transmit it to a new person. But it also means that no blood is getting into the digestive system of the flea, so the flea is starving. And the flea becomes manic about it, like, I'm starving, I'll bite anything, any warm thing I will bite. So that's why it becomes much more aggressive, jumps over to humans and starts biting. Of course, the crowded conditions and rat-infested areas did not help things at all. So what did we learn from the bubonic plague? Quarantine did not work because it wasn't really a human-to-human transmission. It was transmitted mostly by fleas, and since fleas were everywhere during that time, you couldn't really get away from the fleas, even in palaces. But one of the things they did note was that people that did get the bubonic plague and recovered, which was about 50% of the time, didn't ever seem to get it again. So a lot of times it seemed like the pandemic kind of just burned itself out after a big wave of death because almost everybody got it, but then the ones that were left over were immune and couldn't really get it again and therefore continued to participate in the transmission of the disease. So immunity, even though they didn't really know what it was, this idea of getting a disease and then not being real susceptible to getting it a second time, was certainly noted with the bubonic plague, whereas quarantine did not work. Okay, now that we've covered that fun stuff, let's come back to the invention of microscopes. So, as I mentioned earlier, Anton van Leeuwenhoek, a Dutch kind of scientist, he really was a scientist, but it wasn't his profession, invented the first microscopes to be able to view these very, very small bacteria. And he viewed them in his teeth, he viewed them in his saliva, on his skin, in his poop. He was totally fascinated by how many bacteria showed up in his poop. And then one of the other things that he thought was really fun was he would take his teeth scrapings, look at these bacteria, and then drink really hot coffee, and then take the teeth scraping and nothing moved after that. The heat killed the bacteria on his teeth scrapings. Now that's what we know, but he thought it was so fascinating. Coffee kills bacteria. It was the heat from the coffee. Anyway, Anton van Leeuwenhoek was a master glass grounder and made these really great lenses that were able to magnify very, very well and still allow you to see these bacteria. Unfortunately, he was also a bit of a recluse and very, very fussy about his prized possessions. And he was a little bit paranoid and didn't want anybody else to get his microscopes. So when the eminent scientists of the decade came by wanting to have, or at least borrow, his microscope so that they could see these bacteria for themselves, he was flatly, no, go away, I don't ever want to see you again. And so that really hindered the formation of microbiology, because there was only one microbiologist in the world, and that would be Anton van Leeuwenhoek, the only person who could see them. So it wasn't until his death, which was actually in the 1700s, upon his death, his sons started letting scientists have access to his microscopes and therefore share their discoveries with the world, by which point, I'd say the mid-1700s, the existence of bacteria everywhere was pretty well accepted. So, what happened between the discovery of bacteria and when we finally linked bacteria to infectious disease? Because it was not an immediate linkage. In fact, it was 200 years in the making. Now, before we get into this idea of linking infectious diseases to microbes being the cause, let me talk about the importance of that linkage. If I were to ask you what causes smallpox, which you can see here on the left, or syphilis in the middle, or plague on the right, you would say a virus, bacteria, I think, and a bacteria, you just said a bacteria. So those are the causes. But what if I said, no, it's actually voodoo. Voodoo causes smallpox. That's what we think. We think voodoo. Somebody put a hex on you if you get smallpox, or if you get syphilis, or God smited you, or you breathed in bad, noxious air. So if you believe that voodoo causes smallpox, then there isn't any way that you're going to be able to control for that. And so you're not going to enact measurements like quarantine and looking at immunity, or looking for eradicating the cause because somebody put a voodoo on you. That was the cause, supposedly. So what I'm pointing out here is that in order to effectively control and eradicate infectious diseases, you have to know that they're caused by microbes. First of all, that tells you something about transmission. Secondly, it gives you a target to attack with drugs like antibiotics. So if you don't believe that infectious diseases are caused by microbes, you're not going to enact any sort of policy that's going to prevent the spread of that particular infectious disease. Now, why was it 200 years in the making? Well, one reason is because we didn't really understand how life originated. So this is, of course, is still before we understood viruses, but let's just look at some bacterial diseases. In order to understand that bacteria cause disease, and also you get, a bacterial disease from another person or an animal or some other type of bacteria. In other words, the bacteria didn't just come out of thin air, they came from something else. You have to believe what is called the cell theory. Now, the cell theory, or the biogenesis theory, is that all cells come from other cells. But that was not the prevailing theory at that time. The prevailing theory was that life originated spontaneously from a combination of air and water and nutrients. And so we knew this as the theory of spontaneous generation. It was actually, I think, Aristotle that basically kind of came up with this theory, and it just stuck. So even up until the 1600s, the theory of spontaneous generation prevailed. That is to say, if you want like a fly maggot, if you've ever had fly maggots before, little worms, where do they show up from? Air, water, nutrients come together, and voila, you have a fly. Okay, so of course we know that's not how it works, but we needed some experiments to help us disprove this theory that was so grounded into people's consciousness. People really believed this is how it worked. So The first person to disprove this theory was a man named Francisco Redi, and then it was strengthened by another set of experiments, but I'll just credit it all to him to make things easier. 1668, just before Anton van Leeuwenhoek saw his first bacteria. So here's what he did. Basically, people would leave meat out, and then the next day there would be maggots on the meat, and they figured, spontaneous generation, right? So you have air, nutrients, the meat, and if the meat was moist, water. Spontaneous generation, you get fly maggots. And Francisco Redi said, but if I take the meat, fresh meat, and I put it in a jar, and I put cork on top of that jar, there are no maggots. And people said, that's nice, but there's also no fresh air, so therefore we don't really believe that this is not spontaneous generation. The reason why maggots did not spontaneously generate here is because there's no fresh air. So another scientist came and strengthened his experiment by saying, fine, here's some gauze. We'll put gauze over the top of that jar. And now you can still get fresh air in, but the gauze is too small to allow flies to land in. We know, of course, that flies lay microscopic eggs that then turn into maggots. But here, the flies laid their eggs on top of the gauze and hence came forth these maggots. And so Francisco Redi's experiment disproves spontaneous generation, right? Well, kinda. So here's what people got from that. They said, yeah, okay, yes, you've proved it for fly maggots, and so anything that's big, we agree that it must be the cell theory of generation, which means that flies produce flies. In other words, flies reproduce, they make eggs, the eggs generate into maggots, etc., and larger stuff too. Yes, seeds, blah, blah, blah. Yes, big stuff. But that really, really small stuff, the bacteria that Anton van Leeuwenhoek sees in his microscope, all of that stuff is so small, we still think that it's the theory of spontaneous generation. And it wasn't until the 1800s, 200 years later, that the theory of spontaneous generation was finally disproved by a very scientifically prolific man named Louis Pasteur. That will be discussed in the next lecture. Thanks.